Today we will learn the chapter 13, the DNA and its role in heredity. Chapter 1 is composed of 5 subjects, starting from the experiment reveal the function of DNA as genetic material. And we will cover DNA structure, and we will cover how DNA is replicated. So finally, we will cover error in DNA can be repaired. Okay, so this is the first subject is that experiment reveal the function of DNA as genetic material. First one, we call it as the genetic transformation experiment by Griffiths. Okay, first of all, he used two bacteria. First one is wild type. Their space is very smooth. That's why we call it as S strain. The other one is mutant. So mutant, they have their space is rough, is like this. And this is the non-virulent. Okay. So first of all, he did it. For example, the S strain, like living S strain, they are virulent. After we injected into the mice died. How about R strain? R strain, after we inject the living R strain into the mice, but mice is still healthy. Okay, so another one is that just to kill the S strain. Okay, just to kill the S strain by heating. Okay, after that, inject into the mice is healthy. However, so they mixed non-virulent R strain with dead S strain. So they combine to each other. But S strain is killed by heating. But after that, they found that mice dies. So it means that can extract from the dead bacterial cell, especially the S strain, the extract from the dead S strain bacteria, genetically transforming living cells. For example, this living cell is the R strain. Answer is the substance in the heat-killed bacteria transform the bacteria into live and virulent bacteria. So there was a genetic transformation. We don't know. At that time, Griffiths don't know what was the substance. But at that time, he just write down substance in the heat-killed bacteria transform the bacteria into live and virulent. The bacteria is like this. Okay, something is there in the substance. Okay, so after that, another experiment is like this. The question is, what is chemical nature of transforming substances? What is chemical nature? The transforming substance is RNA or protein or DNA. So we don't know at that time. Okay, so at that time, first of all, just kill the S strain by heating. After that, this is they filter this one. So this is the S strain filtrator. S strain yoga egg. Okay. After that, first of all, first group he treated RNase, and the other group is treated protease. So protease is destroyed protein, RNase is destroyed RNA. The final group is add the DNase. The function of DNase destroys DNA. After that, just add this one into the R strain. R strain is non-virulent. After that, they found that in this cluster, virulent S strain and R strain they are mixed each other. However, the final cluster, they only have the R strain. They don't have S strain. So based on this experiment, the answer is the nature of transforming substance is DNA. Okay, so based on this experiment, people say that the transforming substance, Griffiths did not know what is the substance, but based on this experiment, the nature of transforming substance is DNA. 김하은, 이해했어? Okay. Okay, so very important. Okay, so in this slide we will briefly cover bacteriophagy, virus. So bacteriophagy T2, the name of the bacteriophagy is T2. So this is the virus, a species of virus. Their structure is like this. So they have the protein coat, protein. So inside the protein coat, they have DNA is like this. Okay, and this is the this is the microscopic picture. So usually the bacteriophages attach to the space of bacteria. After that, they inject their genetic material. Of that, this red one is the virus DNA. Okay, after that, viral gene take over host machinery. Okay, so virus gene used host cells machinery. After that, they make their own protein coat, and they replicate their own DNA is like this. After that, bacteria is burst, their membrane is burst. Finally, they release about 200 viruses like this. The important thing in this slide is that bacteriophagy has composed of the protein coat, and also they are composed of DNA. But only DNA can be injected into the host cell. Protein coat cannot enter. Only DNA can be transported into the inside of the bacteria. At that, the viral gene used host cell machinery. So after that, they use their own protein coat, and they also replicate their own. The DNA is like this. Finally, they release about 200 viruses like this. But this is the virus life cycle of bacteriophagy T2. The reason why I explain the life cycle is to explain the Hesse-Chase experiment. Okay. The question is, which part of the phagy, DNA or protein, serves as the genetic material? So which part of phagy, DNA or protein? So first of all, what he did. So this is the DNA backbone. So DNA has the phosphate at their backbone. So one group, they radio-labeled this phosphate with P32. Radio-labeled with this phosphate. They used the isotype. So this is phosphate is P32. Okay, they are located in the DNA. So the other group is that in our amino acid, we have methionine and cysteine. Okay, so usually in my protein engineering course, I always ask students to memorize all of structure, but you don't have to do it. The important thing is methionine and cysteine, they have sulfur. They have sulfur like this. So experiment 2, just a radio-labeled protein coat with S35, like this. At that, just impact the bacteria cell with the virus, bacteriophagy. At that, just agitate in a blend to detach virus from the bacteria cell. Just impact it, just 1 or 2 minutes later, just use blender. Okay, agitate it, that's all. After brief agitate, it means that the virus can attach it to the membrane, but after agitate, they will be detached. After that, they did centrifuge. So attach it. After use the blender, they detach it. Okay. After the centrifuge, okay, so in the pellet, there will be bacteria cell. Pellet bacteria is heavier, very heavy, so they are located in the pellet. But people found that P32 is detected in the pellet, not supernatant, not meteor. However, in this case, F35 is not detected in the pellet, is not detected in the bacteria, but indeed detected in the supernatant. Okay, so usually the protein coat is located in the outside. They cannot go inside. Only the DNA can be transported inside, not protein coat. So they were here. So after agitation, the bacteria is detached from the bacteria. But virus is very tiny. They are very small compared to the bacteria. So if the bacteria size is like this, bacteria size is very small like this, very small. So all of them is located in the apt centrifuge, but all of them is located in the supernatant because they are the weight of the virus is very light, so they are located in supernatant. So based on this experiment, result is that phagy with P32 produced progeny phagy which are radioactive. So answer was the DNA carries the genetic information in the bacteriophagy. So based on this experiment, DNA, not proteins, enter the bacteria cell and direct the assembly of new virus. Okay, so DNA, based on this experiment, DNA carries genetic information in the bacteriophagy is like this. Okay. Okay, and let me go to the second subject. DNA has structure that suit its function. Okay, this is the DNA structure. So usually the DNA structure, okay, so we know that Watson and Crick find that they propose the double helix is like this, but actually the 90% of experiment was done by Rosalind Franklin. So Watson and Crick received the Nobel Prize based on their finding. DNA is structure is like the double helix, but actually the 90% of the experiment was done by Rosalind Franklin. Okay. 1% was done by Watson and Crick. Watson and Crick like this, okay? Because the main thing done by Rosalind Franklin, she used X-ray. So after we put the DNA here, after she applied X-ray, after in X-ray film, she got this kind of pattern. But based on this X-ray films result, but she don't know what is this. She don't know. But based on this X-ray films result, Watson and Crick, this structure is only comes when DNA is they are double helix. Okay. Rosalind Franklin provides information about the molecule, but she didn't know that DNA is double helix. But Watson and Crick discovered DNA is double-stranded and the helical structure is like this. That's why. And also, Rosalind Franklin, she did a lot of X-ray experiment. At that time, 9040 and 90PB. We don't know. Nowadays, we know that X-ray is very harmful to people. At that time, they don't know. And she did a lot of experiments without protection. Finally, she got cancer, so she died. Before Watson and Crick received the Nobel Prize, she died. Okay? Okay. Okay, so DNA is the antifederal strand. Okay, so let me start from the basic structure. DNA has this basic. They have sugar. They have sugar. This is sugar. Okay, but they have their second carbon. They don't have OH. That's why this is deoxyribose. They have deoxyribose, and also they have a phosphate at the fifth carbon. They have a phosphate is like this. Sugar phosphate backbone, one sugar phosphate backbone, and the other sugar phosphate backbone. But they combine to each other. So they did a condensation reaction, so they combine to each other. So it means that start from the fifth carbon is end at 3 carbon. Okay, so DNA is usually synthesized from the 5th to 3 direction. So backbone formed through phosphodiester bond, but direction is the 5th to 3. DNA is the different 2 strands, but different strands run in opposite direction. One is start from the 5th to 3. The other one is 5th is start from the bottom. 5th to 3 is like this. Different strands run in opposite direction. So this is Watson and Crick find that based on X-ray crystallography, they proposed that they are parallel 2 DNA strands. They are different strands run in opposite direction is like this. Okay, and at that time, Watson and Crick didn't know that. They know that their structure, they are composed of their antifederal strand, but at that time they don't know. how DNA is replicated. Okay, another 1960s, another scientist found that DNA is replicated semi-conservatively. At that time, there was 3 models. First model is semi-conservative. So this is the original DNA. So this is the daughter molecule following replication. So this is semi-conservative. So semi-conservative produced daughter molecule. Okay. So produced the daughter molecule with original DNA. Okay. So produced daughter molecule with both original is blue is original, and the red is newly synthesized DNA. So finally they will have like this. This is the semi-conservative replication. Another model will be the conservative. So one will become replicated. So it will make the 2 daughter molecules. So another model is the either 2 original or 2 newly synthesized DNA is like this. So this is the conservative replication. Another model is dispersive replication, produced a daughter molecule with a mix of original and newly synthesized DNA is like this. So 1960, we don't know which one is right. But based on Mansell and Starr's experiment, people found that DNA is replicated by semi-conservatively. Let me see some details experiment. So Mansell and Starr, they also used radioisotope. They used nitrogen. So one isotope is nitrogen 15. So usually the nitrogen is the 14. 김서진 알겠어? 14이 오케이 알겠어? But he used another isotope, nitrogen 15, a heavy isotope. Okay, that makes the DNA more dense because the DNA has the base, cytosine, thymine, and uracil, adenine, guanine. DNA, they don't have uracil, they have cytosine, thymine, adenine, and guanine. Every base, they have the nitrogen, like this. Okay. First of all, E. coli was grown in the, nitrogen 15. It means that all of DNA's nitrogen will be nitrogen 15, nitrogen 15, nitrogen 15, nitrogen 15, nitrogen 15, nitrogen 15, nitrogen 15 is like this. After that, then to E. coli was transport to a medium with nitrogen 14. Okay, bacteria were growing in the nitrogen 15. After transfer into the medium with nitrogen 14. Okay, and first group just they sampled at right now just 0 minutes. The other one is 20 minutes. The other one is 40 minutes. Why they used the 20 minutes, 40 minutes? Because of this one, generation time. Bacteria's generation time is 1 become 2 within 20 minutes. What is human generation time? 근역성, what is human generation time? What human generation time? Okay, so baby was born. After that, when they were become around 13 or 14, they start menstruation. It means that they have the ability to have the baby. So this is generation time. Okay, maybe 13 or 14, 15 year. Humans' generation time is 14 or 15. Okay. But bacteria generation time is 20 minutes. Within 20 minutes, one cell become two cell, and two cell become four cell, and that two cell become four cell, four cell. Okay. This is so. That's why they use the 20 minutes, 40 minutes. Okay, 20 minutes means that one cell becomes two. After 40 minutes, two cell becomes four. It's like this. Okay. Okay. At 0 minutes, all of them was very heavy. It's like after that they did centrifuge. Okay. After that, DNA is extracted from the bacteria and placed in the centrifuge. They only put the DNA in the centrifuge. After one group, this is the 0 minute group, all of DNA is 15. They are located like this. Okay, 20 minutes later, this is post generation. They have this band is like this. And 40 minutes later, we find that this one. Okay, so because this is heaviest one, and this is the middle, and this is the light one. Okay, but because after 20 minutes later, we only have the medium range. It means that maybe the semi-conservative will be like this, or this one will be like this, isn't it? However, 40 minutes, we have light one. It's like this. So it means that only semi-conservative model can explain this result. Okay? Dispersed model cannot explain this result. 이해했어? 아주 중요한 건데. 정은이 이해했어? 오케이, 이해해, 알겠지? Okay? Only semi-conservative model can explain this result, okay? That's why based on Messerson and Starr's experiment, people found that DNA is replicated by semi-conservatively. Okay? Okay. And also, let me explain the origin of DNA replication. So DNA is replicated semi-conservatively, and also in DNA replication, in prokaryote and eukaryote, there is a big difference between the DNA replication in prokaryote and eukaryote. For example, this is the prokaryotic chromosome. Their shape is round, so we call it as plasmid. Okay, they only have one origin of replication. Okay, after that, 20 minutes later, one becomes two chromosomes. However, we have a eukaryotic. Our eukaryotic have multiple, lots of origin of replication. This is the main difference. Okay, because our genome is very large. So if we only have one origin of replication, it will take a lot of time to replicate our DNA. But because we have lots of original replication, so the replication time is very short. Okay. Okay. But prokaryote is only replicated within 20 minutes. But in human, we don't know exactly, but maybe 24 hours, we don't know exactly, but larger than 20 minutes, okay? Longer than 20 minutes, okay? So in this slide, I will explain the DNA replication process, starting from the RNA primer. Okay, after that, I will explain the replication in the lagging strand and the leading strand. Finally, I will cover telomerase, is like this. Okay. So maybe some of them already take the biology course, advanced biology course during their high school. I think some of them already know that, but I will explain it, okay? I will explain it very simply, okay? So this is the DNA strand. Usually DNA is separated for replication. So we will see that later. But after separation, it becomes a single strand, okay? But they need this kind of primer, RNA primer. So usually replication comes from 5' to 3. Okay, so at that time there was no primer, but premase. Premase bind to the template strand and synthesize the green colors RNA primer. Okay, just the RNA primer size is about 10 base pair or 20 base pair. Okay, after synthesis of RNA primer is complete, premase is released. After that, DNA polymerase is bind to here, synthesize the new DNA is like this. But I will teach you some detailed information. Okay, this is the DNA polymerase. DNA polymerase is bind to the DNA template strand, DNA strand. Okay, so DNA polymerase structure is like the right-handed. Okay, this is DNA polymerase, right-handed. This looks like finger, thumb. Okay, and this looks like finger. Okay, this is we call it as palm, 손바닥, palm. Okay. DNA polymerase is shaped like open right-handed. At the bottom, they are closed, but in the upper, they are open. Finger, this finger, especially this finger, recognize DNA. And the palm, 손바닥 has the enzyme, has the active site. DNA polymerase is an enzyme, so they have an active site. After that, they synthesize the DNA molecule in the palm, 손바닥, okay? Okay, like this, okay? Like this, okay? DNA is synthesized. So, okay, let me see some detailed information. First of all, DNA helicase. Okay, so I will teach you the exact procedure. First of all, DNA helicase bind to the double-stranded DNA. After that, it unwind. So it will make the single strand is like this. So usually the single-stranded binding protein single-stranded binding protein bind to each strand. So without this protein, they'll combine again. But if so, single-stranded binding protein keep the template strands separate. They are separate. That they have the leading strand, they have a lagging strand. That DNA polymerase is bind to that they elongate it like this. First of all, helicase and single-stranded binding proteins. At that, primase bind to at the end. So they make RNA primer. At that, DNA polymerase bind to here. So they synthesize, they replicate the DNA is like this. Okay, so this is the leading strand. How about the lagging strand? We will see that. So leading strand is that synthesis of leading strand is continuous, is like this. Okay, at the first time they continue, they start from here, like this. They are continuous. How about the lagging strand? Okay, lagging strand. We call it as Okazaki fragment. Okazaki fragment. The important thing is DNA is only replicated from the 5th to 3 direction. 5th to 3 direction. Okay. So in the lagging strand, first of all, so this is the 5' DNA. This is means that another opposite will be the 5th 3'. But DNA cannot be replicated from the 3 to 5' direction. So in only cases, RNA primer is bind to here. At that, they synthesize DNA from 5' to 3' direction. Okay, at that, helicase open more. At that, RNA polymerase. At that time, RNA primase cannot bind to here, but they are open is like this. Now is the RNA primase bind to another location. At that, DNA polymerase bind to here, so they make the DNA strand is like this. So this is the Okazaki fragment. Why we call it as Okazaki? Why do we call it as Okazaki? So Okazaki was a Japanese scientist. So Okazaki and his wife, they studied this study in the America. Husband and wife scientist. But to find this kind of thing, they used a lot of isotype, radioactive material. The Okazaki husband dies. And based on the husband's experiment, and the wife finished. At that, they found this phenomenon. That's why this is recorded as just a Okazaki fragment is like this. Because Okazaki find this phenomenon. Okay. So once again, this is a lagging strand story. We call it as a lagging strand story. Leading strand is very simple. However, in the lagging strand, okay, usually the primase forms RNA primer. Okay, so primase forms RNA primer. At that, primase removes that DNA polymerase bind to here. So they will synthesize a replicated DNA is like this. So DNA polymerase 3 as, so this is, there is no nucleotide, but DNA polymerase 3 add nucleotide to the fragment until it encounter the primer on the previous Okazaki fragment. Okay. After they go to here, they finished. After that, this DNA polymerase will be detached from the strand. Okay, this is the lagging strand story. Okay. After that, so initially they have the RNA primer is here. Now is another polymerase. We call it as RNA polymerase 1. So they removed RNA primer. They have the ability, DNA polymerase 1 has the ability to replicate, but they have another ability to remove the RNA primer. So DNA polymerase 1 hydrolyzed the RNA primer. After that, replicate it with the DNA. But however, the final, each fragment, they have final gap. At that time, DNA ligase bind to here. They ligate the final step. Is like this. Okay, this is the lagging strand story. So in midterm exam, okay, so what is the lagging strand story? Explain the detailed procedure of lagging strand story, starting from the pre-mase. Pre-mase formed RNA primer. After that, DNA polymerase 3. We have to write down pre-mase DNA polymerase 3. After that, DNA polymerase 1. Finally, DNA ligase. Okay, so this is the lagging strand story. So another component, we call it as sliding DNA clamp. The full name of this component, sliding DNA clamp. Sliding, they are sliding shoe like this, sliding. Okay, so usually this one, okay, this one bind to the next to the DNA polymerase. Okay, the main function of this sliding DNA clamp, this one is increased efficiency of the polymerization, the DNA replication. Because by keeping the RNA polymerase bind to the strand, so it means that so the DNA polymerase, so if the DNA polymerase cannot bind to here, okay, after replication happens, sometimes they can be dissociated. This time it happened, there was no replication here. However, this sliding DNA clamp bind to the next to the DNA polymerase. So it means that DNA polymerase does not have to repeatedly bind to template and substrate. Because keeping the DNA polymerase 3 bound to the DNA substrate. 박수빈 이해했어? Okay. So what is component to increase the efficiency of DNA polymerization? What is it? Sliding DNA clamps. Okay, there will be some question in the midterm exam. I can ask you. 단답형? Simple question. You have to write down sliding DNA clamps, okay? So finally, we will learn the telomere and the function of telomerase. Okay, so this is the chromosome structure. They have 2 arms and 2 legs, and the center we call it as centromere. So at the tip region, end of chromosome, we call it as telomere. So usually at the end of the chromosome, their DNA structure is composed of GGG, ATT, GGG, ATT, GGG, ATT, GGG, ATT. The telomere are made of repetitive sequence, repetitive sequence. The main subunit is GGG, ATT. The GGG, ATT, GGG, ATT, GGG, ATT, GGG, ATT. So this repetitive sequence can protect chromosome from damage. It looks like the cap, 안전 cap. Okay, it looks like cap. Okay, it protects the chromosome. Okay, so at the end, so for example, after DNA replication happens in the leading strand, and this is the lagging strand. They make the Okazaki fragment. It's like this. But at the end of the lagging strand, okay, so there will be primer, RNA primer. The terminal, so at the end, this sequence, finally the RNA polymerase is removed. So this will be the blank is like this. So this region cannot be replicated. So it means that after DNA is replicated one time, so it will this fragment cannot be replicated. It means that DNA is shorted one time the replication more shorted like this. The DNA will be shortened after replication. But it is not happen because of telomerase. Telomerase. So telomerase, okay, so at the end of the lagging strand, telomerase is bind to at the end. The telomerase itself has the RNA template. They have RNA template itself. Okay, so telomerase bind at the end. Okay, so because they have RNA template, for example, so DNA can be replicated from the 5' to 3. So this region can be replicated like this. Okay. Also, this region, they have the blank region. So after that, telomerase move to the end. So this one is moved to the end. They move to the end. After that, this one is the 5', 3'. So 5' to 3', this gap will be filled. Okay. So telomerase moved to the end. At that time, DNA polymerase is the synthesized DNA here and also synthesized here from the 5 to 3 direction. So this process can be repeated multiple times to lengthen the telomere region. So it means that even though DNA is replicated, DNA can maintain their length. They cannot be short like this because of telomerase. Okay. So first of all, we learned that at the initial step, helicase After single-stranded binding proteins, after pre-mase make the RNA primer is like this. So in the leading strand, you can after DNA polymerase 3 bind to here, that they synthesize from 5' to 3 direction. How about in the lagging strand? Okay, we learned the lagging strand story. Okay, so pre-mase form more RNA and DNA polymerase add new nucleotide. After that, RNA polymerase hydrolyzed, they removed RNA primer. After that, they also replicated the DNA. Finally, at the gap, DNA ligase joined the Okazaki fragment. Okay, but at the end, and also another sliding DNA clamps helps increase the efficiency of DNA polymerization. At the end, they will find the telomere region. But usually the telomere region should be shortened like this, because this region cannot be replicated. However, telomerase was there, so telomerase they have the RNA template itself. So this region can be replicated, and also this region can be replicated is like this. Okay. Now we will cover fourth subject, errors in DNA can be repaired. Okay, so if people becomes older and older, old, old person has higher percentage to get cancer, isn't it? Because of this one, DNA polymerase make mistake. During the DNA replication, DNA polymerase 3 or DNA polymerase 1 replicate the DNA, but they did mistake. That's why people has the mutation in the DNA. Okay, and people, okay, for example, old person they did the loss of DNA replication. The mutation is one mutation is accumulated, 2 mutations, 4 mutation, 5th mutation. After 10th mutation, they got cancer. But for example, 근혁성 is about 24 hours, 24, how old are you? 20 pips. Okay, but because he only have 1 or 2 mutation. Okay, that's why he did not get cancer yet. but the important thing is that old person have higher chance because of the DNA mistakes, DNA polymerase mistake. Usually the one long base out of the 100,000 base is replicated. 100,000 base pair is replicated. Usually the one base is the mistakes is happen. So usually there are 3 mechanisms: DNA proofreading, also mismatch, or excision repair is happen. So old person has higher chance to get the cancer, but some old person still have no cancer because of the DNA proofreading. We have we have this ability. Our human body has DNA proofreading ability. Okay, for example, DNA polymerase 3 itself he did this one did a mistake, but this one also has DNA proofreading ability. So during the DNA replication, incorrect nucleotide may be added to growing chain. But DNA polymerase 3, they can proofread. Okay, proofread. What is proofread? In Korean, what is proofread? 점검한다는 거죠, 맞죠? 점검. 다시 점검한다는 거죠. DNA polymerase 3 has the ability to proofreading. So immediately excise this nucleotide. After that, added correct nucleotide is like this. That's why even the old person, some old person, they don't have cancer because of they have this kind of ability. DNA polymerase 3 has this kind of ability. Another one is mismatch repair. So usually this during replication, nucleotide was mispaired, okay? Long nucleotide was added. At that time, our human body has mismatch repair protein. So mismatch repair protein recognize this region. After that, this remove this region. Okay, excise the mismatched nucleotide, and some adjacent nucleotide is like this. Okay, after that, DNA polymerase 1. DNA polymerase 1, not 3, DNA polymerase 1 added correct nucleotide. Finally, DNA ligase, the fragment is like this. Repair the remaining nick is like this. So this is mismatch repair. Another is excision. Excision repair. Okay. So usually the excision repair, a nucleotide DNA is damaged. For example, if we receive a lot of UV, so sometimes if we receive during the summertime, if we receive a lot of UV, at that time our DNA is damaged. Because UV has the very short wavelengths, they have high power. Sometimes DNA can be damaged. Okay, for example, mismatch means that in this case, DNA polymerase 3 has typically, they did mistake one long base out of 1,000 base pairs. Okay, so that's why mismatch repair is happened. But sometimes, several kinds of UV, sometimes DNA can be damaged. So this nucleotide or next nucleotide can be damaged. At that time, another protein, excision repair protein, they scan it, they found it, this region. After that, they remove it. After that, DNA polymerase 1 added a new nucleotide. Finally, DNA ligase repaired the ligase. Okay? So this is the 3 mechanism. Okay? Do you know that? Okay.